When I was in graduate school, we used to joke that when you were earning your PhD, you were basically learning more and more about less and less. And there's more than a grain of truth in that. But at some point, learning more and more about less and less is about the only way to move forward. Well, hello, and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis for well on my way to 40 years. I continue to keep up to date with the latest research that's going on, knowing full well that there's no cure just around the corner, or maybe even close, but hopeful that there will be one in my lifetime. If not a cure, at least something that will make some of my most irritating symptoms go away. And I remember from way back in the 1980s when the MRI, the magnetic resonance imaging machine, came online, and we first were able to look at a living brain in real time and see things that we had never seen before. We were finally able to see lesions, especially when using the contrast tool. You could see the lesions light up so that you knew what was active and where it was and how many and how large, etc., etc. You could track that over time. Never before had we been able to do anything like that. And it has really changed the way we treat multiple sclerosis. Many of us are on a disease-modifying therapy that would never have been possible if we hadn't been able to use the MRI to see what was happening inside our brains. But we've been at that now for quite some time, and even though MRI machines are getting more powerful, better and better, and doctors are becoming more and more familiar with what they're seeing, at some point, you kind of need to know more than that, because MS is still incurable, intractable, and annoying. So now we got to look at this a little differently, I'm thinking, and the best way to do that is to see what's going on at a more granular level than what they're able to see with the MRI. And lo and behold, that is indeed what is happening. They're beginning to look at the microstructures of the brain, so that they can figure out what's causing the problems that we see with multiple sclerosis. Now, I want to share a couple of articles with you that I've just recently seen on this topic. But before I do that, I want to show you a video that just gives you a little bit of an idea of how we even identify them, what is the process, how it's done. I know it's a couple of years old, but it should work just fine for this purpose.
Today we're discussing something pretty incredible, and that is the world's most detailed brain scan. Scientists at the Massachusetts General Hospital in Boston have produced the world's most detailed MRI scan of the human brain. The scan took 100 hours to complete, producing an image that can see detail as small as 100 micrometers. To put that in scale, that is about the size of the thickness of a single human hair, a scale unprecedented in human MRI scanning. Previously, to see this level of detail, the brain had to be dissected and placed under a microscope. This has issues with the development of artifacts, which simply means a distortion of the tissue so it doesn't reflect normal anatomy. This makes it difficult to draw conclusions of the microstructures within the brain. However, with this scan, it is possible to see incredibly small details without having to disturb the brain tissue. The brain was donated for scientific research by a 58-year-old lady who died of issues unrelated to her brain. Therefore, the brain was presumed to be completely healthy tissue. A live brain could not be used, because in order to scan to this level of detail, the brain had to be kept perfectly still for over 100 hours. This, the researchers felt, was a big ask for a living person. In order to scan the brain, a special container containing urethane had to be made. This was designed to keep the brain very still, as any movements would create artifacts, rendering the scan useless. In addition to this, as is the case with any tissue kept for a significant length of time, air bubbles will inevitably develop. The container therefore also had to allow for these air bubbles to escape so it wouldn't affect the scan. Now we understand the complexity of a process such as this. Let's see the scan itself. Wow, that is absolutely incredible. The details that can be seen throughout the brain are just stunning. The differentiation between the white and grey matter, as well as the details in the cerebellum, are truly mind-blowing to see. Unfortunately, a full detail of the scan cannot be appreciated due to the limitations in the resolution of the video. A scan of the human brain of this detail could allow researchers to unlock the secrets of many poorly understood disease processes. It's fair to say that scans of this quality will help revolutionize our understanding of key processes within the brain.
Well, I hope you found that interesting. But let's go ahead now. We'll take a look at some of the articles that are looking specifically at microstructures in a way that will hopefully lead us to some new information, new knowledge about multiple sclerosis. Well, I found this first article on the Springer Link website, and it is entitled "Brain Microstructure is Linked to Cognitive Fatigue in Early Multiple Sclerosis." And it was published in March of 2024. And if we go down here, look at the authors. All of them are from the University of Liège in Liège, Belgium. And according to the abstract, we don't get the whole paper here, but that's okay. I think we'll get the gist of it just from the abstract. Cognitive fatigue is a major symptom of multiple sclerosis from the early stages of disease. This study aims to detect if brain microstructure is altered early in the disease course and is associated with cognitive fatigue in people with MS compared to matched healthy controls. Recently diagnosed persons with MS, and there were 18 of them, that's what the N means, N equals 18. They had 18 subjects, and their age was under 45 years old, with either a relapsing remitting or a clinically isolated syndrome course of the disease, and healthy controls, 19 of them, matched for sex, age, and education, were analyzed. Quantitative multi-parameter maps, MTSAT, PD, R1, and R2, and don't ask me what those are, of persons with MS and healthy controls were calculated. Parameters were extracted within the normal appearing white matter, cortical gray matter, and deep gray matter, respectively. Bayesian t-test for independent samples attest between group differences in brain microstructure, while associations between score at a cognitive fatigue scale and each parameter in each tissue class were investigated with generalized linear mixed models. Patients exhibited lower MTSAT and R1 values within the normally appearing white matter and the normally appearing gray matter, and higher R1 values in normally appearing dark gray matter compared to healthy controls. Cognitive fatigue was associated with PD measured in every tissue class and to MTSAT in normally appearing white matter regardless of group. Disease-specific negative correlations were found in persons with MS in normally appearing white matter, R1 and R2, and normally appearing gray matter, R1. These findings suggest that brain microstructure within normal appearing tissues is already altered in the very early stages of the disease. Moreover, additional microstructure alterations, e.g. diffuse and widespread demyelination or axonal degeneration, in persons with MS may lead to disease-specific complaint of cognitive fatigue. Well, I think that the most important sentence here, at least for me, was what they say right here. These findings suggest that brain microstructure within normal appearing tissues is already altered in the very early stages of the disease. Doesn't Dr. Gavin Giovannoni say the same thing? That all MS is progressive MS? That we start doing this progression independent of relapse activity right from the get-go. And it doesn't have to be associated with big lesions that show up on the MRI. There's this kind of smoldering MS that goes on. And this seems to be right in line with that. Looking at the microstructure of the brain is giving us some important insights into how MS works. And I think that is going to be key in curing MS. We have to really understand what it is that we're trying to cure. I think that what we're seeing a lot of with the disease-modifying therapies is an attempt to treat basically the visible signs of MS. But if there's a lot going on that we aren't seeing, looking at these microstructures is going to give us some very important insights into how MS progresses. But you may well ask, well, how are we going to look at every single microstructure in the brain? There's gazillions of them, right? I don't know that humans would be able to do this in any kind of a time frame that most of us would like to see happen. Fortunately, one thing computers are really, really good at is detecting patterns. And so I just saw this article. The paper just came out very recently from NYU, New York University. The article is entitled Artificial Intelligence Tool Detects Sex-Related Differences in Brain Structure. This came out on May 14th, 2024. And it says that the findings highlight the need for diversity in neurological and psychiatric research. Well, what I like about this right off the bat is that what the findings that they've already been receiving from this artificial intelligence tool are telling them that they're going to need to do things a little differently, at least as far as clinical trials and the conclusions that they draw from them. They're going to need more diversity in those trials than they tend to get. And I have shown that in a number of the trials that we've looked at, that most of them seem to be weighted toward women because they know that MS is more common in women than in men, but not all of them. Well, let's take a look at what they're actually trying to get at. But first, let's admire this lovely art rendering. It says all the models correctly identified the sex of subject scans between 92% and 98% of the time. And it's a Getty image. I don't know if it's based on any kind of test results or anything, but I just like it. It's a lovely image, so enjoy that. But as we go on, we'll look at the abstract. Artificial intelligence, or AI, computer programs that process MRI results show differences in how the brains of men and women are organized at a cellular level, a new study shows. These variations were spotted in white matter, tissue primarily located in the human brain's innermost layer, which fosters communication between regions. Men and women are known to experience multiple sclerosis, autism spectrum disorder, migraines, and other brain issues at different rates and with varying symptoms. A detailed understanding of how biological sex impacts the brain is therefore viewed as a way to improve diagnostic tools and treatments. However, while brain size, shape, and weight have been explored, researchers have only a partial picture of the brain's layout at the cellular level. Led by researchers at NYU Langone Health, the new study used an AI technique called machine learning to analyze thousands of MRI brain scans from 471 men and 560 women. So this is a slightly larger study than many, or maybe a greatly larger study than many. The study that we just looked at had a sample size of 18 people with MS and 19 healthy controls. So this is considerably larger than that. Results revealed that the computer programs could accurately distinguish between biological male and female brains by spotting patterns in structure and complexity that were invisible to the human eye. The findings were validated by 3 different AI models designed to identify biological sex using their relative strengths in either zeroing in on small portions of white matter or analyzing relationships across larger regions of the brain. Our findings provide a clearer picture of how a living human brain is structured, which may in turn offer new insight into how many psychiatric and neurological disorders develop and why they can present differently in men and women, said study senior author and neuroradiologist Yvonne W. Louie, MD. Dr. Louie, a professor and vice chair for research in the Department of Radiology at NYU Grossman School of Medicine, notes that previous studies of brain microstructure have largely relied on animal models and human tissue samples. In addition, the validity of some of these past findings has been called into question for relying on statistical analyses of hand-drawn regions of interest, meaning researchers needed to make many subjective decisions about the size, shape, and location of the regions they chose. Such choices can potentially skew the results, says Dr. Louis. Well, I hate to say that, but I think they would, because humans have bias in things that they think and do and say, whether they want to or not, whether they even realize it or not. Anytime you make a decision, you're going to have bias. And hopefully, whoever programmed the AI tool has screened for that pretty carefully. We're going to hope so anyway. The new study results, published online May 14th in the journal Scientific Reports, avoided that problem by using machine learning to analyze entire groups of images without asking the computer to inspect any specific spot, which helped to remove human biases, the authors say. And this is exactly the kind of thing I was just talking about. You're trying to eliminate the possibility that someone will make a decision that will, whether they mean it or not, will influence the results, will influence even the things that are examined in the first place. For the research, they say, the team started by feeding AI programs existing data examples of brain scans from healthy men and women, and also telling the machine programs the biological sex of each brain scan. Since these models were designed to use complex statistical and mathematical methods to get smarter over time, as they accumulated more data, they eventually learned to distinguish biological sex on their own. Importantly, the programs were restricted from using overall brain size and shape to make their determinations, says Louis. And you notice they put in quotes here a couple of the words, smarter and learned, because they don't want us to get the idea that these AI tools get smarter or learn things the way humans do. It's a bit different. It's quite a bit different, actually. But all it's doing is very similar to the texting feature on our smartphones, where after a while it learns the words that we're likely to use, and as soon as we type the first couple of letters, it gives us the words as suggestions, thinking that this is probably what you mean. And I have to say, they do. They get smarter and smarter, if you want to use that term, all the time. By repetition and refining, these programs are quote-unquote learning to make better choices. According to the results, all of the models correctly identified the sex of the subject scans between 92% and 98% of the time. And I'm telling you, that's pretty good. I think that a human doing this would take a lot longer and wouldn't be any more accurate, if even close. Because we're talking about very, very small microstructural differences. It's hard to detect those. And the vast amount of microstructures in the brain is just daunting. And so people have tended to limit the region of the brain that they look at. Well, by making that choice, they may be unwittingly ignoring a part of the brain that's very important to this investigation, and using an AI tool will prevent that from happening. Several features in particular help the machines make their determinations, including how easily and in what direction water could move through brain tissue. These results highlight the importance of diversity when studying diseases that arise in the human brain, said study co-lead author Junbo Chen, MS, a doctoral candidate at NYU Tandon School of Engineering. If, as has been historically the case, men are used as a standard model for various disorders, researchers may miss out on critical insight, added study co-lead author Varalakshmi Bayanagari, MS, a graduate research assistant at NYU Tandon School of Engineering. Bayanagari cautions that while the AI tools could report differences in brain cell organization, they could not reveal which sex was more likely to have which features. She adds that the study classified sex based on genetic information and only included MRIs from cisgendered men and women. According to the authors, the team next plans to explore the development of sex-related brain structure differences over time to better understand environmental, hormonal, and social factors that could play a role in these changes. Funding for the study was provided by the National Institutes of Health, and there's the grant numbers, as well as by the United States Department of Defense grant number, and you can read the grant number. Interesting that the Defense Department has a stake in this, and my antenna are up on this one. I'm kind of thinking, okay, is that just because they're interested in the AI tool? I have a feeling that's probably why. They want to see if they can apply this AI tool to other problems that are defense related, one would think, but one never knows, does one? And now that we've looked at that, I want to show you one more article. It doesn't pertain directly to MS, but it talks again about microstructures and the ability to see things we hadn't seen previously. And so I do believe that we're looking at the future of MS diagnostics and treatment. And maybe it will lead us a little bit closer to that all-elusive cure. Let's check it out. And I couldn't resist. This is so cool. I decided that I would show you this article that I saw in Nature just this month, in 9th of May 2024, this was published. Cubic millimeter of brain mapped in spectacular detail. Google scientists have modeled a fragment of the human brain at a nanoscale resolution, revealing cells with previously undiscovered features. And here is an artist rendering. Pretty nice. That looks like nice wallpaper background for your desktop, doesn't it? In fact, I might even think about doing that. But here's the caption on this. Rendering based on electron microscopic data showing the positions of neurons in a fragment of the brain cortex. Neurons are colored according to size, and we give credit to the Google Research and Lichtman Lab of Harvard University. And the rendering is by D. Berger, also of Harvard University, and quite talented. This is just lovely, actually, and just to think about it, it kind of looks like a very, very busy circuit board. And that's what's going on inside our brains all the time. Maybe not in such beautiful color coding, but one never knows. And they say that researchers have mapped a tiny piece of the human brain in astonishing detail. The resulting cell atlas, which was described today in Science and available online, reveals new patterns of connections between brain cells called neurons, as well as cells that wrap around themselves to form knots, and pairs of neurons that are almost mirror images of each other. The 3D map covers a volume of about 1 cubic millimeter. So that picture you just saw, that hardly covers any distance at all. That hardly covers any of the area of our brain. 1/millionth of a whole brain, they say, and contains roughly 57,000 cells and 150 million synapses, the connections between neurons. It incorporates a colossal 1.4 petabytes of data. It's a little bit humbling, said Viren Jain, a neuroscientist at Google in Mountain View, California, and co-author of the paper. How are we ever going to really come to terms with all this complexity? And as they say, it's only a sliver of the brain that they've been able to image so far. The brain fragment was taken from a 45-year-old woman when she underwent surgery to treat her epilepsy. It came from the cortex, a part of the brain involved in learning, problem solving, and processing sensory signals. The sample was immersed in preservatives and stained with heavy metals to make the cells easier to see. Neuroscientist Jeff Lichtman at Harvard University in Cambridge, Massachusetts, and his colleagues then cut the sample into around 5,000 slices, each just 34 nanometers thick, that could be imaged using electron microscopes. Jane's team then built artificial intelligence models that were able to stitch the microscope images together to reconstruct the whole sample in 3D. I remember this moment, going into the map and looking at one individual synapse from this woman's brain and then zooming out to these other millions of pixels, says Jane. It felt sort of spiritual. And yeah, I mean, look at the complexity. Now this is not an artist's rendering. I think this is the real thing from the images that were taken. According to the caption, it says a single neuron in white, shown with 5,600 of the axons blue that connect to it. The synapses that make these connections are shown in green. Now, looking at this, you have to realize some incredible things. We know that with advanced MS, we've lost quite a few of these neurons. They're more than damaged, they're just gone. But when you think that this is just a very, very tiny fragment of your entire cortex, and even smaller of a portion of your whole brain, you recognize that losing a few doesn't damage you to the point where you can't even function because you've got so many other ones. And then there's that wonderful function of the brain, neuroplasticity, that allows the brain to just keep trying different routes to get the same job done once it can't use the old route. As they say, when examining the model in detail, the researchers discovered unconventional neurons, including some that made up to 50 connections with each other. In general, you would find a couple of connections at most between two neurons, says Jane. Elsewhere, the model showed neurons with tendrils that formed knots around themselves. Nobody had seen anything like this before, Jane adds. The team also found pairs of neurons that were near-perfect mirror images of each other. We found two groups that would send their dendrites in two different directions. And sometimes there was a kind of mirror symmetry, Jane says. It is unclear what roles these features have in the brain. But before this imaging was done, they didn't even know these were there. So think of all the promising developments that will come of the new knowledge that they have. I'm sure hoping that it will anyway, because pretty pictures are nice, but what we really want is a cure for multiple sclerosis. As they say, they need proofreaders because the map is so large that most of it has yet to be manually checked, and it could still contain errors created by the process of stitching so many images together. Do you think maybe AI could help with this? Maybe we need to let them know that there's this tool that's available. Hundreds of cells have been proofread, but that's obviously a few percent of the 50,000 cells in there, says Jane. He hopes that others will help to proofread parts of the map they are interested in. I'm assuming he's talking about actual qualified people, not just people like us. As much as we might like to help, I think we have to leave this one to the professionals. The team plans to produce similar maps of brain samples from other people, but a map of the entire brain is unlikely in the next few decades, he says. As I gathered from what was said about the brain tissue that they were able to obtain from someone who was getting surgery for epilepsy, it must have been a part of her brain that they actually removed. And I don't think most of us are quite that philanthropic that we really want to donate our brains while we're still alive. So I don't know the details of where they're going to come up with the rest of the samples that they need, but it's nothing we can get in a hurry over. But like I said, if they could use the AI tool to help with some of this proofreading, wouldn't that be nice? This paper is really the tour de force creation of a human cortex data set, said Hongqi Zhang, director of the Allen Institute for Brain Science in Seattle. The vast amount of data that has been made freely accessible will allow the community to look deeper into the microcircuitry in the human cortex, she adds. Gaining a deeper understanding of how the cortex works could offer clues about how to treat some psychiatric and neurodegenerative diseases. This map provides unprecedented details that can unveil new rules of neural connections and help to decipher the inner workings of the human brain, says Yang-Soo Kim, a neuroscientist at Pennsylvania State University in Hershey. Well, I know this doesn't seem to pertain to us immediately, but I'm hoping that you're getting the idea that things are moving along, and there's quite a lot of excitement from a lot of different quarters, not just one research organization, not just one scientist, not just one country. They're looking at this from many directions all over the world. Sooner or later, somebody is going to hit on some connection. I think we've already got a lot of the building blocks, but we're not there. Still, I think it's time to be a little bit patient and hopeful. But in the meantime, I think we just need to keep ourselves as knowledgeable of what's going on as we can, so that we know that we're not crazy to be hopeful. And also, of course, our major responsibility is to keep ourselves as healthy as we possibly can, so that when these breakthroughs come, we are able to take advantage of them. But that's all I have for you for now, so I'm looking forward to seeing you again in my next video.